Hey, good evening to one and all. My name is Madhukar. I am studying BSc-BZC first year TTWRDC means both at Adilabad. Today my topic is structure of bacterial cells. This is the structure of a bacterial cell. And context, introduction, cell morphology, structure of bacterial cell wall, plasma membrane, extracellular external structure, intracellular internal structure. Introduction. All bacteria are unicellular organisms that reproduce by binary fission. Most bacteria are capable independent metabolically extends and growth, but special of chlamydia and rickettsia obligatory intracellular organisms. Bacteria are extremely small and are most conveniently measured in microns, 10.6 meters. The bacteria cells are usually between 0.4 and 1.5 micrometers in short diameter. Cell morphology. Bacteria have a characteristic shape. The comma microscopic micrologies are spiral or ovoid. Koki, rod-shaped bacilli, comma-shaped vibrio. Some Koki characteristic group in pair or chain, some from graphe-like cluster or of spherical cells, some round cocciform cubic facet. Bacteria are of the species or separately. The microscopic appearance is therefore valuable in the classification of diagnosis. This is the structures of coccus, round shape, bacillus, rod shape, and streptococcus, chain shape, diplococcus, chair shape, spirocheta, spiral. Structure of a bacterial cell. This is side one is a structure of bacterial cell. The protoplasm, the whole body of a living material. Protoplasm is bonded peripherally by a very thin elasticity and semi-permeable cytoplasmic membrane. Outside and the closely covering this lie the rigid supporting cell wall, which is portions of relativity are permeable. Next one, the cell wall. The cell wall encases the protoplasm and lie immediately external to cytoplasm membrane. It is 10.20 meter nanometer thick, strong, relative rigid through with some elasticity and openly porous, being freely permeable to solute molecules smaller than 10 kDa in mass and 1 nanometer diameter. It shows granules structure and lacks microtubules. Gram positive and gram negative bacteria have a different types of bacteria cell wall, and this is the structure of a cell wall. Next one, gram positive of cell wall. The gram positive types wall relatively thick, about 30 to 100 nanometer, and it generally has a simple unformed appearance under the electrons microscope. Some 40 to 80% of a wall is made through complex polymorph polydoglycan. In these types of wall, succulents consist of a multilayered peptidoglycan which during growth, development, and inside the outside mechanisms. Next one, covalent bond peptidoglycans are compounds such as tichos acid, typical substituted polymers, glycerol phosphate, or ribitol phosphate. In some bacteria, Mycobacterium, the wall contains lipid, while in other strain of Streptococcus contains carbohydrates. The compositions of the wall can vary with growth condition. For example, bacillus. The availability of a phosphate affects the amount of cell wall touches acids. This is the structure of gram-positive cell wall. Gram-negative cell wall. The cell wall of gram-negative bacteria are more chemical complex, thinner and less compact. Peptidoglycan makes up only 5 to 20% of a cell wall and is not a outermost layer. The peptidoglycan or gram-negative bacteria is located between the plasma membrane is an outer LPS membrane. This LPS membrane is similar to the plasma membrane but is less permeable, is composed by liposaccharide LPS. And this is the gram-negative cell wall structure. Plasma membrane. Plasma membrane in bacteria composed of phospholipids contains a polar ground attached to a 3 carbons glycerol backbone. There are also 2 fatty acid chain dangling from the outer carbon of glycerol. Phospho, the end of nucleus hydrophilic, is attached to water. The fatty acid hydrocarboxylic membrane is also contains protein. Next one, typical of a 30% of a membrane. Most of them are placed in the membrane, so that hydrocarbonic acid associated with the lipids in the membrane and hydrophilic ammonic acid are outside membrane interacting with either the cytoplasm. Functions of the plasma membrane is selective permeability that help control what moves into outer of the cell wall, and this is the extracellular structure. Fimbriae or filli. Fimbriae or filli are elongated or hair-like proteinaceous structure which project from the cell surface. They are special in those gram-negative cell. Fimbriae are typical 22.3 nanometer diameter from 0.1 micrometer to several. They may occur all over the cell or may be localized. Flagella. The flagella are made up of a class of linear protein cell flagella. Flagella which arise to various points on the cell surface. Flagellated bacteria are described as monotrichous, locotrichous, or ferritrichous. It's depending on how the flagella arranged. This is the structure of a capsule, and this is called two names: slime layer or capsule. The capsule of a most bacteria consists of a polysaccharide. The bacteria of a single species can be classified into different capsules. Several structures based on the fine chemical structure of this polysaccharide. Intracellular structure plasmid. The term plasmid was first introduced by the American molecular biologist John Lederberg in 1952. The plasmid is short, usually circular double standard segment of DNA. The found in the cytoplasm separate from the main bacteria chromosomes. Their size varies from 1 kbp to over 100 kilobase pairs. Plasmids are capable autonomous replication. Plasmid can transfer gene one genes from one cell to another cell. Ribosomes. The cytoplasm contains a large number of low high molecular weight substance RNA and approximately 2000 ribosome cells. Bacteria have 17 ribosomes comprising 30s and 50s subunits. Functions of a ribosome. Ribosomes are the organelles for portion synthesis. Mesosomes. Mesosomes are a convoluted or multinucleated membranes bodies visible in the electron microscope. They develop by complex integration of cytoplasmic membrane into the cytoplasm. Functions. The functions in compartment of a DNA at cell division and at sporulation, having a function of analogous to the eukaryotic cell, providing a cell membranous supporting for respiratory enzyme. Nucleids. The nucleids is a region of a cytoplasm where chromosomal DNA is located. It is a not a membrane bonded nucleus but simple an area of the cytoplasm where the strand of DNA are found. Most of bacteria having a single circular chromosome that responsible for replication. Although a few species do have two or more smaller circular auxiliary DNA strands called plasmid are also found in the cytoplasm. So thank you for the listening class. Thank you.